faithfully delivering value and interpreting great books from around the world.
Hello, this is Sally talking books.
Today, I'm interpreting the book The Formation of Life, subtitled How the Human Body Builds Itself from Scratch. The human body is an incredibly intricate device. Where do you think the most awe-inspiring part of human design shows up? Feel free to share your thoughts in the comments below. And an adult's body is made up of nearly 1 trillion cells. That's over 10 times the number of stars in the Milky Way. This complex system has hundreds of cell types, each perfectly placed, dividing and renewing just right to keep life running smoothly. How do we go from a single fertilized egg to a whole person? Where do I actually come from? How is a brand new life formed from nothing? What developmental principles guide the human body as it builds itself? The Formation of Life will give you the answers. The author is Jamie Davies, professor of experimental anatomy at the University of Edinburgh in the UK. He's dedicated his life to studying how mammalian organs build themselves, publishing over 90 research papers and several academic books. Jamie also serves as a researcher at the Royal Society of Medicine and the Higher Education Academy, and as editor-in-chief of the Journal of Organogenesis. In this book, Jamie shares discoveries and the latest findings from human histology research, taking us back to life's beginning, through those first 9 months, to understand the extraordinary journey from a single cell to a complete person. The author uses the painting process as an analogy for the embryo's full development. I'll walk you through the book's main content via the 3 stages of development. Stage 1, sketching the outline, where a fertilized egg develops into the basic form of a baby. Stage 2, adding details, refining the functions and tissues of body systems. Stage 3, fine-tuning, building systems to adapt to the outside world. First, let's dive into what the fertilized egg goes through in the sketching stage. Developing into the rudimentary form of a baby, human development starts with a fertilized egg, about 0.1 millimeters wide, just visible to the naked eye, and thousands of times larger than a regular cell. Now, the fertilized egg doesn't pause for cell growth. It keeps dividing to form a multicellular entity, taking that crucial first step in development. So, at this stage, the fertilized egg divides just like regular cells. We've all covered cell division in high school biology class. First off, a DNA molecule copies itself precisely into 2 molecules, using strict nucleotide pairing and enzymes. Then, chromosomes, made of DNA and proteins, line up neatly right in the middle of the daughter cell. And finally, contractile proteins make the cell membrane pull inward and split cleanly into 2, forming 2 new daughter cells. So, how does the cell figure out where the center of those new cells is? And how do chromosomes get split perfectly between the 2 cells? The cell manages to pinpoint the center and divide everything evenly.
For chromosomes, the key players are microtubules. These tiny proteins form around an organelle called the centrosome, which has a special enzyme that helps link microtubules into long tubes. The microtubules stretch out from the centrosome until they hit the cell membrane, creating forces. Shorter ones pull harder, longer ones pull softer. So the centrosome gets pushed to the center, balancing everything out.
Figuring out the centers for both daughter cells works similarly. Microtubules from 2 centrosomes push against each other, and the centrosomes just naturally drift to where the centers will be. No need for knowing the cell's shape or any coordinates. Cells organize themselves using simple rules. Microtubules shooting out from the centrosomes set the center and make sure chromosomes split apart properly, so every new cell gets a full set. But microtubules are unstable. They break down after a while. But if they latch onto specific proteins, they stay put for longer. We call those microtubule binding proteins. Every chromosome has a spot with these proteins. Microtubules pop up and fade away randomly. If a microtubule bumps into that chromosome spot, it sticks around. So, the centrosome hooks up to chromosomes with microtubules and heads to the center. to prevent duplicate chromosomes in each new cell. It's all about controlling microtubule stability. The replicated chromosomes are connected by a special protein complex. When chromosomes are pulled apart, the protein complex senses mechanical tension and releases signals to stabilize the microtubules. Conversely, it releases signals to rapidly degrade the microtubules. This mechanism ensures that the replicated chromosomes are accurately distributed to the two daughter cells. The process of cell division perfectly illustrates the developmental principle of adaptive self-organization, where simple components following simple rules can collectively form large-scale, intricate structures. By sensing mechanical forces through microtubules to determine the center, and by leveraging microtubule stability, chromosomes are cleverly divided equally among daughter cells, ensuring each gets a full set. Ooh, well, This mechanism is repeatedly reused in fertilized egg division. Early cells use nutrients inherited from the mother for rapid division, completing each in about 10 to 12 hours until forming a 64-cell embryo. At this point, the cells are as small as possible and urgently need nutrients from outside for growth, so some must specialize in acquiring nutrients for others. Thus, the cells in the embryo begin to differentiate and become distinct from each other. These cells formed from fertilized egg division are identical. How do they decide their developmental direction? Actually, the answer isn't complicated. The embryo can derive developmental cues from geometric patterns. After the fertilized egg divides into 64 cells, some are completely surrounded by others and are inside. While some cells have part of their membrane facing external space, bathed in the fluids of the fallopian tube and uterus, cells have a complex mechanism to sense if their surface is facing external space and use this information to decide their next developmental step. Cells partially exposed activate previously silent genes, developing into the earliest differentiated tissue, the trophoblast, which provides nourishment to the rest of the embryo. Once the trophoblast matures, it starts pumping nutrient fluid into the embryo. Part of the inner cell mass, which was originally enclosed, contacts the fluid, senses signals, and develops into the dense hypoblast, which later forms supportive tissues like the yolk sac, providing blood and nutrients to the fetus early on. Ooh! The remaining cells develop into a new layer, the epiblast, which will form the entire fetus. After hypoblast formation, genes and central cells activate to synthesize DNA-binding proteins. These cells migrate to the hypoblast edge and cluster together. Their secreted proteins diffuse short distances, influencing epiblast development. In the epiblast, areas near the cluster develop into the baby's head, while distant regions unaffected by the proteins become the lower body. The embryo transforms from a radially symmetric sphere to an oval structure called the primitive streak. Sometimes the primitive streak cell mass doesn't remain single but splits into two clusters. Each cluster develops into a separate life, the most common origin of identical twins. Over 70% of twins form this way. If activated hypoblast cells fail to cluster, the epiblast may form two primitive streaks, developing two individuals. This rare twin formation, less than 1%, is dangerous as streaks lack clear boundaries, often causing failed separation. Resulting twins often share body parts, what we call conjoined twins. After primitive streak formation, the epiblast develops into three distinct layers: endoderm, gut lining and digestive glands, mesoderm, and ectoderm. This gastrula stage embryo, mesoderm forms bones, muscles, blood, lymph tissues, while ectoderm develops into skin and neural tissue. From fertilization to gastrula takes approximately 15 days, after which nervous system development begins. A key process is neural tube closure. Along the embryo's midline, the notochord secretes signaling proteins activating ectoderm genes. Activated cells sync, merge laterally, and detach to form the neural tube, foundation of the nervous system. Neural tube closure in humans occurs around weeks 3 to 4 of pregnancy, and it's a process prone to complications. If closure fails in the brain or spinal cord, children may be born with developmental defects like paralysis or incontinence. Scotland historically saw frequent cases of unsuccessful neural tube closure. Follow-up studies revealed ectoderm cells grew too slowly in these cases, preventing tube edges from meeting. Further research showed folate levels in neural tube tissue are critical. Adequate folate ensures proper ectoderm cell proliferation, promoting tube closure. In early industrial Scotland, pregnant women lacked folate-rich foods like produce, causing high neural tube defect rates. Today, prenatal folate supplements are globally adopted to prevent neural tube defects. While the nervous system develops, the endoderm simultaneously forms digestive pathways. So, in about 4 weeks, a single cell embryo self-organizes using simple rules into thousands of cells with basic structure. The embryo now has clear head-tail orientation, neural tube along the back, digestive tract extending forward. With recognizable basic structures sketched, the next phase refines intricate details. Part 2. Let's explore how embryos add details to build complex tissues. Human survival requires simultaneous, even conflicting processes. For example, a mother digests proteins while synthesizing new ones for breast milk. The body solves this through compartmentalization. Specialized organs divide labor. Lungs oxygenate blood, intestines absorb nutrients, each with tissue subunits. The pancreas secretes digestive enzymes first, then hormones to regulate absorption. Kidneys filter waste. Developing organs, viscera, limbs, neural networks is the embryo's detail-adding mission. The heart is the earliest organ to form, beginning development around day 19 of pregnancy, primarily from the outer edge of the mesoderm in the head region. And these cells differentiate into cardiogenic cells, attracting other myocardial cells to fuse and develop into the heart. The lungs, liver, pancreas, and gallbladder originate from intestinal branches formed by the endoderm. These branches receive signals from mesoderm cells, continuously proliferating and refining to develop complete organs. Meanwhile, the spleen, gonads, kidneys, and urinary system derive entirely from the mesoderm through tube merging, aggregation, and cellular development. During early embryonic development, cells rely on random molecular diffusion for short-distance nutrient exchange. As cell numbers increase, nutrient transport becomes inefficient, necessitating an efficient blood system to deliver nutrients throughout the body. Oh, I can't. The blood system comprises 4 components. First, the liquid component, carrying most nutrients and toxins.
Second, oxygen-carrying blood cells floating freely in this liquid.
Third, a closed tube system delivering blood to tissues. Fourth, a heart providing propulsion, circulating blood between nutrient-supplying and nutrient-utilizing tissues. Around day 14 post-fertilization, the blood system begins developing from mesoderm cells. Human vascular network development continues throughout life, with capillaries responding rapidly to tissue oxygen demands. When tissue cells experience oxygen deprivation, they secrete hypoxia-inducible factor, HIF protein. This signal prompts vascular wall cells to rapidly grow vessels toward the most oxygen-depleted areas. Fresh blood delivers oxygen to hypoxic tissues. Once oxygen levels normalize, HIF degrades rapidly, stopping the hypoxia signal. Limbs are essential for human survival. The earliest signs of limbs appear around day 24 of pregnancy, when cells beneath the ectoderm receive mesoderm signals and rapidly proliferate. Small protrusions form on the embryo's sides, continuously proliferating and differentiating into bones, muscles, and other tissues, requiring efficient blood supply during growth. If blood vessels fail to grow in time, the fetus may develop shortened or misformed limbs. For example, starting in 1958, many pregnant women took thalidomide to suppress morning sickness. Originally, it was primarily used as a sedative and anti-inflammatory drug. However, in 1961, scientists discovered that a molecule produced when thalidomide breaks down in the body strongly inhibits fetal blood vessel growth. If mothers took this drug during fetal limb development, the risk of limb shortening or malformation increased significantly. People are always curious about fetal gender. Within 7 weeks of pregnancy, gender can only be determined through chromosomal testing. But the real core determining gender is the gonads. Gonads mainly consist of two cell types, germ cells and somatic cells. Germ cells originate from the primitive streaks, epiblast, migrate via the gut's outer surface to the gonad area, and fuse with somatic cells to form gonads. Embryos with a Y chromosome produce specific signaling proteins that activate silent genes in gonadal somatic cells. These cells rapidly multiply, secreting hormones to develop male characteristics. Sure, without a Y chromosome, different hormones are released, developing into a female. The process involves complex mechanisms where gonads receive DNA signals, secrete hormones, and regulate development of reproductive organs, mammary glands, bone structure, and hair distribution. Among fetal organs, the central nervous system is most astonishing. Neural networks connect billions of cells, coordinating all tissues and organs. The nervous system begins developing during weeks 3 to 4 of pregnancy. The central nervous system originates from the neural tube. Its head expands and folds into the brain, while the remainder becomes the spinal cord. Head neurons connect to all body tissues and organs. Neurons mainly connect through elongated structures. Dendrites receive signals and perform local computations. Axons transmit neuronal signals via neurotransmitters to other neurons and muscle cells. Axons can extend indefinitely as needed, up to tens of thousands of times the neuron's body length, to form new connections. For example, axons connecting the spinal cord to feet reach about 1 meter. Nervous system connections develop throughout life. Through learning, neural networks refine connections, increasing complexity and efficiency. Embryos add detail through cell communication, proliferation, and differentiation. And ultimately, the fetus must leave the uterus and adapt to external environments. But finally, let's explore how embryos fine-tune systems to develop post-birth learning and adaptation abilities. After birth, babies encounter sensory chaos, sounds and sights they must interpret to form consciousness. In 1960, Canadian neuroscientist Donald Head proposed learning's mechanism, modifying synaptic strengths between neurons. For example, dogs naturally salivate when seeing or smelling food. If we repeatedly pair an unrelated stimulus, like a whistle, with food, the dog eventually salivates to the whistle alone. This occurs because connections between sound processing and salivation neurons strengthen. Without interference, the dog completes learning through conditioning. Modern AI biology parallel simulate this biological mechanism, strengthening connections via data training. But synaptic modification isn't learning's only mechanism. New research shows neural connections can also be pruned or rewired. Visual system development illustrates this. Prenatal retinal brain connections are crude. Axons connect haphazardly, some correct, some erroneous, resulting in blurred infant vision. After birth and opening their eyes, babies' brains start reshaping connection patterns based on how visual neuron synapses fire consistently with receiving cells. If a neuron connects only to axons from the left eye, since the left eye shows the same field of view, those signals should be pretty consistent. If some axons keep sending signals that don't match others, it means they're hooked up wrong, and those faulty connections get destroyed. The free axon ends then try to link up with other cells, repeating this over and over until they connect right, changing how things are wired. By constantly tweaking retinal neuron connections, the brain links consistent inputs from the outside world, having them control the same neurons. This change in connection strength and reorganization lays the groundwork for advanced skills like language and association. This helps babies keep learning and adapting in the wild, ever-changing world after birth. When babies leave the womb to grow their minds out here, they've got to deal with microbes, some good, some bad, so they got to learn to make friends and fight foes. First up, making friends. These pals are probiotics in the gut. In a healthy gut, each gram of tissue holds billions to tens of billions of bacteria. They do key jobs like secreting enzymes that break down big molecules the body can't handle into small ones the gut can soak up and use. Oh.
They also break down toxic stuff fast, making food safer.
Babies can pick up probiotics through the birth canal or during feeding. These probiotics enter the baby's mouth, hitch a ride to the gut via swallowing, and kick off a win-win partnership with human cells. For instance, Bacterio-D-thiothiomicron, a common gut bug that breaks down big carbs into small sugars, secretes a molecule detectable by small intestine cells once it lands in the gut. After getting the signal, small intestine cells add fucose groups to sugars, so Bacteroides thiota omicron gets plenty of food. Other microbes that can't make fucosebases starve because they can't get to the food. Every part of the digestive tract uses similar communication to befriend probiotics, creating ideal environments for specific bacteria they need. But not all bacteria are friendly. Countless bacteria and viruses eagerly seek chances to invade our bodies.
Babies must learn to fight back.
Fortunately, humans have developed powerful defenses that block and eliminate most invaders. Our defense has three layers. 1. Passive barriers like skin's dead cells, mucous membranes, and perforins in bodily fluids. 2. Active defense by mobile phagocytes. Phagocytes detect bacterial waste signals and migrate toward invaders. After capturing bacteria, they release signals, increasing blood flow to bring more phagocytes.
They also release toxic compounds to destroy bacteria, though this damages healthy tissues too.
Increased blood flow causes local redness and heat. Toxic attacks stimulate nerve endings, causing pain, are common inflammation symptoms. Though annoying, inflammation serves a purpose.
Some bacteria hide inside cells where phagocytes can't reach. Destroying nearby tissues stops disease spread.
3. The adaptive immune system, unique to vertebrates.
When facing new pathogens, B and T lymphocytes activate, multiply, and specialize to eliminate threats.
Activated B and T cells retain rapid response capability against specific threats, preventing reinfection with lasting protection. Vaccines harness this mechanism to build lasting resistance against specific pathogens. In the fine-tuning phase, the body interacts with unpredictable environments to quickly, adaptively reorganize the nervous system, building mechanisms to befriend and fight microbes, ensuring healthy growth after leaving the room. The formation of life is a complex process. As the famous British poet Samuel Taylor Coleridge said, "The 9 months before birth might be more fascinating than the 70 years after." On the other hand, the grand truth is simple. A fertilized egg follows adaptive self-organization principles to gradually differentiate and develop into tissues and organs. What other simple and elegant operational laws like this exist in nature? Feel free to leave comments below and share the formation of life. That's the end of our interpretation for you. Listen notes are below the audio, and we'll see you next time.